In a previous video on between sample normalization, I mentioned a strategy that relies on the postulate that the mRNA within a sample, that proportion, that total amount, would be the same across various samples, right? It would be the same for the samples. Hopefully you took at least triplicates from the untreated group and hopefully the triplicates from the treated group, and that each one, you just extracted that exact right amount of mRNA, the same amount of mRNA or total RNA. If you're looking at, RNA transcripts besides RNA, mRNA. That is, for certain few cases, a reasonable approximation. And I mention approximation because it's never going to be exact. But in other cases, in many other cases, it's an unreasonable assumption to make, especially when the sequencing depth is out of line or you used a different sequencing prep for the technology, even just how long you ran the sequencer. There's a lot of different factors that can sort of mess up that assumption. So why were strategies such as FPKM and TPM originally developed? They aren't exactly very effective in terms of between sample normalization, and that's sure they were originally designed to handle a different problem, which is called within sample normalization. Within sample normalization, that seems pretty confusing from the outset, right? Why would you need to normalize within a sample if you use the same sequencer, if you use the same technology, if you use the same sequencing prep? It even came from the same sample, right? The same cells, hopefully. Well, it actually solves a rather different problem. As you see here, between the isolation and the preparation step, there's a certain process called fragmentation that occurs. In essence, you see these sort of long fragments, you see these short fragments that I've already denoted. You can include the poly A tail in this. These sort of get broken down or cut into pieces that will be specific fragment lengths. So the original transcripts that you have are turned into these sort of more standardized fragments. Now the fragment length is not the same for every fragment, but it follows a distribution, which depends a lot on the sequencing prep that you're using to prepare the RNA for sequencing. And this is because you're taking, only 150 base pair reads, 100 base pair reads, whatever your design is, and it doesn't make sense that if you have a really long RNA, 2,000 base pairs, that you would only sort of get the first 100 or so. It makes more sense to cut it up and try and extract as much, information about it as possible. But this fragmentation process introduces its own sort of artifacts, and this is what within sample normalization tries to fix. So let me just sort of add on that fragment length follows, as I mentioned, a distribution. And just for a couple of rough examples, you could have a mean of 200 base pairs, or a mean of 400 base pairs, or 600 base pairs. That could be common. Or a standard deviation of 50 base pairs, or 100 base pairs. Those can also be common. So just to have a bit of an idea what is typical or common. In terms of these fragments, they get sequenced, and what you end up with from the sequencer, once it's sort of aligned, once you have your FASTQ files and you align them, is you get counts. And the counts represent the number of fragments aligned to a particular section of the genome. So number of fragments aligned. And you can take any one particular. If you look in sort of the integrated genomics viewer, you can see exactly how those fragments are stacking up against the specific reference genome that you're using for the alignment. But here is where the sort of artifact, or here is where the problem, you might assume, right, that these counts represent the counts of particular genes. You have, one gene and there are 12 counts, and you have another gene and there are 5 counts. And so you say, okay, there's 12 counts of one, 5 counts of the other, so they exist within the cell. They exist within the cell in a 12:5 ratio. For every 12 of gene A, there are 5 of gene B. Theoretically, could, sourcing it perhaps from the sample of an untreated. But in fact, this would be a overinterpretation, overanalysis of what these counts actually mean. What's important to keep in mind is sort of how these fragments are sourced again. And I'll do a quick example to make that clear. Let's say we had a gene in blue here, and we also had a second in green. Now these would be sort of transcripts of the gene, whatever the transcript is that exists within the cell. And now we go ahead and we want to do fragmentation, and we say, okay, fragmentation occurs and it's broken up into all of these different pieces. So the blue one's broken up into 1, 2, 3, 4, 5, 6, 7, 8, 9 parts. Whereas the green one almost doesn't make it, but let's say it's broken up into 2 parts. Now it's time to prepare the library. You add the specific adapters on and you sequence it. This one will have a count of 9, or pick up a count of 9 that picks up all the transcripts. Let's assume you do enough, you have enough sequencing depth. You're going to find everything essentially. And this one will only show up 2 counts. But does that mean that there are 9 versions of the transcript for every 2 versions of the green transcript? 9 versions of the blue transcript for 2 versions of the green transcript? No, there's 1 green transcript in the cell and 1 blue transcript. And so they exist within the cell in this 1-to-1 ratio. But the way that you picked it up on the sequencer is that you found 9 because the blue transcript is much longer and can fragment into many more pieces, can start in many more places. And so looking at this, you see that the main factor at play is the length. And this is ultimately what the point of within sample normalization is. It's to try to understand what you might term actual relative abundance. So how do these specific within the cell, how do these things actually coexist? Or to what ratios, in what ratios do they coexist? And what you can imagine how that's important within a biological context to understand relative existences of link RNAs and proteins, and you can start to hypothesize, how they interact. Is it a 1-to-1 ratio? Potentially thinking about structural hypotheses, etc., etc. So actual relative abundance. And so basically, these strategies oftentimes use length scalings, lengths of these, based on the lengths of these transcripts that are, usually known. And these are incorporated within the formulas for FPKM and TPM. We'll get into more specifics about how these formulas work, in future videos. Thank you for listening.